you can turn in your ticket towards a membership, and about 2.5 admissions to After Dark covers a membership, and that covers 50 Thursday nights of programs, including the next 5 of these talks that are in your brochure tonight. So a little bit about the format tonight. We're going to try something a little bit different. Dr. Muotri has brought something very special tonight with him. He's brought some cortical organoids for you to look at. And so we're going to look at those as part of the program. What we're going to do is we're going to hear from him for about 30 minutes. And then he'll be joined on stage by my colleague Rob Semper, who's our senior scientist in charge of research and development here at the Exploratorium. So a little bit about Dr. Muotri. He's a geneticist originally, and then went into neuroscience, and he's going to tell you a little bit about that himself. We thought he was really interesting to bring to you because he asks questions and then he's making a tool to answer it. So these organoids are crazy compelling. They are alive, and they're both an instrument and a new set of questions. I want to just say one more thing before I talk about the program a little bit more. The Biofutures series is a part of a project that created I should say we are celebrating a new collection of exhibits on the floor in our living systems section, which is towards the back of the museum in Gallery 4, and I really hope that you take a chance to look at it before you leave. There's about 24 new exhibits, I think, and they are full of living cells and cultures, and they celebrate the fact that everyone is a self and we're all made of cells. And I want to just thank Christina Yu, who's the director of the development of the new collection, who's in the back, for the creation of this new set and the support of this new series. I want to thank Rob Semper, my colleague, for joining us tonight. And he'll be paying attention to what Dr. Muotri says and have a bunch of questions for him at the end. So hopefully you'll stay and enjoy that conversation with us. For now, please join me in welcoming Dr. Alysson Muotri.
Okay, I think this is on. And can I have my first slide, please? All right, okay. Thank you so much for the invitation. It's my pleasure to share a little bit of our research that we're doing in the lab. I'm actually from San Diego in California, where I have a stem cell biology lab, and we work on conditions like autism or schizophrenia, and we try to understand how those conditions actually happen in the brain and if there is ways to fix that. But before I jump into what we are doing in the lab, let me tell a little bit on how I get involved with neurosciences. So I'm a geneticist by training, so I have a PhD in genetics, and I was kind of looking for opportunities. And one day I was invited to talk to the European Parliament about my work. And after the talk, there was a bunch of people in the room talking about what are the biggest challenges of our world. So this is where we live, and I have a challenge for you. Think about what you think would be the biggest problem that we are facing nowadays. So if you're like me, you are probably thinking about one of those, and that's what those people were thinking as well. But defining the problems is just the beginning. I think another huge challenge is how to fix it. And I have a challenge for you. I mean, how are we going to fix them all those problems? How there is only one way to fix all those problems. So just pause and imagine, how are we going to fix all those problems? So that was the exercise that I did, and I realized that the only way to solve those problems is to use the human brain. So there is no other species who's going to save this planet. Is the human brain who's going to save this planet. So when here we have a conundrum with the human brain neuroscience. Most of the neuroscience today, I mean, we work on trying to understand the human brain as it is. So we put subjects on MRI scans, and we analyze their brain waves, and we try to understand how the brain works. But the brain is such a complex machine that we are just observing that complexity, and we don't know how the brain gets to that stage. So we turn into animal models. There's a bunch of my colleagues who study mice and try to understand how the human brain works, studying the mouse brain. But here's the thing. The mouse brain is so tiny, it fits in my eyeball. So it's millions of years of evolution that changes those two brains. So good luck trying to understand the human brain by looking at the mouse. So we really need a model, a human model, to really understand how the brain is formed. And just to give you like an idea of how little we know, I mean, if you all realize that this is a car, because all the pieces are very familiar to you. If I break the brain apart and put all the pieces like that, there is no neuroscientist today that will tell that this is a brain. Because we just don't know all the parts. We don't know how it's formed. So we have no idea. And you might ask, so, I mean, why don't you learn how to do it? And here is the biggest challenge. The human brain is formed inside a uterus. And we don't have access to that human embryo brain. It's going to be unethical to do an experiment with a healthy human brain. So we just don't study. Instead, we rely on non-invasive technologies such as ultrasound to kind of realize how the brain is going, but we don't fully understand or appreciate how the first neuron fire, how the first networks are formed and turn into the complex brain that we all have. So that challenge has been a major roadblock for all neuroscientists because we really can't understand the human brain. So that's why we turn into models. So when I give talks to lay public, I realize that people have a problem with the word model. And I'm from Brazil. When I go back and talk to my fellow Brazilians and they ask me what you're doing, and I said, oh, I use stem cells as a model to understand the human brain. And I said, what do you think it's a model? And I get all kind of crazy stories. I mean, people think that this is a model, right? And I said, wow, you're right, this is a model. So, I mean, a child might think that this is a model of a train or a flying machine is also a model. And everybody is right, because a model is an imperfect representation of the reality. So this very simplified representation of the reality are models that we all use, and the best models are the ones that you can increase in complexity to get closer to the reality, which is the human brain. So I mentioned to you that we use the human brain tissue, mostly post-mortem tissue, as a model, but the cells are dead, so we cannot hear from a single neuron or how the networks are formed. We study cells from individuals such as blood, but blood don't form synapses, so they are not the brain cells that we want. And again, I mean, most of us actually turn into mouse models. And again, I mean, there's millions of years of separation between those species. So what my lab has decided to do is to use stem cells to recreate the brain from scratch. And you probably hear from the media that what we're doing is we have a mini brain in a dish. And this is the image that most of you might have. And I'll tell you that that's not what we have, and we have it right here. So you can see it exactly how it looks like. So the word mini brain is misleading because we don't have a fully formed brain there. Instead, what we have is something way more simpler. And there is a bunch of limitations to this model. I'll list them to you before I continue. For example, most of those neurons that we can form are immature neurons. these what we call brain organoid, that would be the correct scientific term. It's a brain organoid, are not vascularized, so they cannot grow that big. We don't have all cell types represented. We don't even know if we're growing them in the right condition. We're going to see here that they're floating in a solution that we think or we optimize for the growth of those human neurons, but we actually don't know. It's empirically defined. So there's room for improvement. And so my lab over the past 5 years have created a recipe to generate these brain organoids that is fully optimized and very simple way to do it. So we start with human pluripotent stem cells. These human pluripotent stem cells are similar to the human embryonic stem cells. And where do we get them? Well, the embryonic stem cells you get from the human embryo. So couples that go to clinics for in vitro fertilization, they donate the leftover embryos, and we can extract these embryonic stem cells, and we can use for this purpose. But we have a more powerful way to get those cells, because we can get those cells from anybody who is alive. So it means that if I take cells from my skin, or my hair, or my dental pulp, I can go back to my lab, reprogram these cells to make these pluripotent stem cells. And you know what happens? You capture the genetics or the genome of the person where the cells were derived from. So the donor cell type will carry your genome here. And when I make this protocol to create these brain organoids, guess what? I'll have a brain organoid from you because it contains all your genetic information. So this would be interesting in the future when I talk about disease modeling. I'll get there. But so the protocol is very simple. It's a series of repetition. You just grow the cells and let them self-aggregate in 3-dimensional. They will self-aggregate, start to migrate out, and form different brain structures. You can actually guide them to the different types of brain cells that you want to have in there. So this is a protocol for a cortical organoid. We mostly focus on the cortex because that's the region where we see problems in conditions such as autism, schizophrenia, dementia, Alzheimer's. So it's a very important brain region. So what you have at the end is a brain organoid that in a couple of months you start to have this cortical plate. So this is more or less what brain organoids look like. This is a postdoc in the lab, a plate with 6 wells, and each white dot in those wells is one of these brain organoids. So they can grow up to 0.5 centimeter, so you can actually see them at naked eye. And once I make a batch of brain organoids from someone, I don't only end up with 1 or 2, I end up with hundreds, if not thousands of them, so I can use for different experiments. So the way this happened, I think that's the most interesting way. It's all genetically programmed. The cells do what they're supposed to do. So I hope you can see here that there are cells migrating out from the core on top. So this is one of the brain organoids that we just placed in a 2-dimensional structure. And I hope you can see that there are some cables. This is what we call radioglia cells, where the progenitor cells migrate out to form the cortex. So in a 3-dimensional structure, this would be from inside out. That's how your cortex is formed. That's how your brain is formed, from inside out. So if you slice one of these organoids, what you have is a structure like that, as a ventricular zone in the center where the progenitor cells are located. And as they migrate out, you're going to have the formation of the cortical plate. So the cells that we have in there are different cell types. This is just one way to identify all the cell type you have. Glutamatergic neurons, these are the first type of neurons that appear during human development. These are excitatory neurons. We have glia cells. These are cells that support the formation of synapses. GABAergic neurons, these are your inhibitory neurons. They appear a little bit late during development, and they'll help to fine-tune the networks. And finally, your pool of progenitor cells that continues to produce these neurons and glia cells, so you can have more cells in these organoids. We can keep them for several years in the lab, and I'll tell you why this is not a good idea. But these progenitor cells, they will kind of stop growing, producing more neurons when they reach about 1 year. And mostly because they are not vascularized, so there is no nutrients going inside of these organoids, so they stop proliferating. So the good news is that we have all the cell types that we need for the brain to work. All the major cell types are there, and we have the perfect anatomy. We have a ventricular zone, we have the cortical plate. As soon as scientists start to realize that we have that tool that mimics human embryonic development, we start asking, can we use this tool for something useful? And I'll give you one example from my lab. I was, in 2015, a colleague of mine called me and said, have you ever heard about the Zika virus? And I never heard about the Zika virus before, and said, oh, we think that it's causing some kind of macrocephaly outbreak here in Brazil. But we don't know if that's the causal agent or if it's something else. So what we did was we collect a little bit of Zika virus and we expose to these brain organoids, and we see that the brain organoids shrink. So that was the first causal evidence to suggest that the Zika virus was leading to macrocephalic cases in the northeast of Brazil. We couldn't do that with mouse models because the mouse brain developed so fast, the Zika virus didn't have the time to kill the cells so we can see a macrocephalic brain. So you need the slow developing human brain to model a condition like that. But most importantly, once we figured out that the Zika virus was killing the cells, we could use that model to try drugs that could block the action of the virus. And 2 years later, we found a drug called sofosbuvir. It's a drug that's already approved by the FDA for another virus. And this drug was able to block viral replication. So right now, if we have another Zika virus outbreak, and there is a woman who is pregnant and it's positive for the virus, she can take this drug and the baby will be safe. So this is one of the few cases where we have the right tool and the right biological question that we can use to solve and understand the virus biology and provide a solution for the problem. We were so happy with that idea that we decided to expand that idea to everything else that is exposed in uterus. So we think that the uterus is just like an incubator, right? I mean, it's just growing the babies in there. But it's not. It actually shapes who you are. And women that are pregnant, they are exposed to thousands of chemicals. Some of these chemicals might not be good for the developing brain, and unfortunately, there is no way to know because all the toxicology is done in mouse brains. So if the mouse brain does not recapitulate that, we're going to miss it. So you probably heard about Tylenol. Increases the chances for you to have a kid with autism or other neurological disorders. So why we took so long to realize that Tylenol was bad for a pregnant woman? Because it needs to catch up with the epidemiology. So those kids have to grow up and receive the diagnosis so we can do the correlation with the use of Tylenol. So we think that we can use this model now to create what we call a brain-safe label. So everything that goes in uterus, we can test and put a red flag so we can investigate better or block if those medications or drugs goes into the body of a pregnant woman. So that's one of the applications. The other application that I think was really cool, and this is a recent work, we can take some of these brain organoids and we can seed them with glioblastoma cells. Glioblastomas is one of the nasty cancer types that really kills children. So here what you see, these big balls are brain organoids, and the green cells are glioblastoma that we took from a patient. This patient could not respond to any drug. So we use this model to find a potential treatment that would wipe it out all the cancer cells and leave the brain intact. So the good news here is that if we did it for one person, we can do it for everybody. and this can be a personalized treatment. So you can have your own mini brains with your own cancer, and we can figure out what kills your cancer cells and leave your brain intact. So this is slowly moving into clinics, and we're gonna get there. So all those applications were so nice because we have these brain organoids, and again, I mean, the shape of these brain organoids and the cell types really mimics the human developing brain. But there was one thing that we never had. None of those brain organoids actually function like the human brain. So we could never had neurons firing in a synchronized network as the human brain does. And we think this was the case because the protocols we had was not that good. But as we improved the protocols, we thought that perhaps we have a chance. And the way to measure those networks is to place some of those brain organoids on multi-electrode arrays. So these are dishes where you have the electrodes printed in the bottom, and you place the organoid in there, and you start recording from their activity. So you have activity maps like that, or raster plots where each channel will have different traces representing when neurons fire. So you can record over time. So when we start doing that, I was, to be honest, not very excited, and I'll show you why. So if I plot a brain activity here over time, you can see that most of the work is below 5 Hertz. And these are human neurons that we used to have. Or even though if you can keep them a long time, up to 32 weeks, you never pass this 5 Hertz. But we want to get to this 20 Hertz that is close to the primate brain. So it means that there is more neurons active and the brain is just firing in a frequency that is more close to the human brain. So now I'm going to show you how this new protocol works and how it changes everything. So what you see is weekly measurements of these organoids over time. And you can see that when they reach about 25 weeks, they start to peak in activity and becomes almost exponential, reach close to that 20 Hertz. So that was unprecedented. We never imagined that this will happen and make us really happy. But also open the possibility to start seeing if those brain organoids will actually show neural oscillations. So neural oscillations are these waves that you can record once you have electrodes through your skull. So this is what we call electroencephalogram, and it's easy to do, easy to record, and it's important because those brain waves correlate with different states of being. So if you're paying attention, your brain wave is going to have certain shape. If you're sleeping, your brain wave will have a different state. If you have a seizure, your brain wave will dramatically change. So they correlate with different diseases state, different developmental time. And there's lots of studies correlating these neural oscillations with human behavior. The one that I like the most is when they put these electrodes in a group of people watching a movie, and they start recording all at the same time, and all the time when there was a social interaction in the movie, a dialogue between two people, the neural oscillations from everybody in the audience synchronized. Is that amazing? So it tells how much social our brains are programmed to behave. So we are social beings. So our brainwaves synchronize when we are talking to our friends. So it's much higher chances to synchronize with a familiar face than with an unfamiliar face. So that's why it's important to have these brain waves when I'm studying autism, for example. So, and we do see these brain waves in these organoids. That was the biggest surprise. They actually appear when they are about 4 months of age. By 6 months, you see that they are very synchronized, and by 8 months, they become highly complex. So that complexity, it's interesting because it says that the networks are getting in a stage where they become very plastic. And plasticity in the brain or in the network is something desirable because it makes you change ideas, makes you make different interpretations of the world. So once we have these waves, we start asking, what is the age of our organoids? How can we compare these brain waves that we are recording from these structures to the human brain? And since this starts from very early embryonic stage, and I told you that we cannot place electrodes in a developing human brain, so we have to rely on something else. We have to rely on babies that are born prematurely. And what happens when a baby is born premature? You place some of these electrodes in their heads just to monitor their overall brain health, brain activity. And what we learn by doing that is that as we develop, we pass to a state that's called trace discontinue. It means that in the brains of these babies, you have a peak of activity followed by a quiescent, a silent time, and another peak of activity. So you can see those peaks of activity over time. So that's why we call trace discontinue. As we grow older, and this happens to everybody, the period between the activity, the silent quiescent time, becomes shorter and shorter and shorter, so our EEGs or our brain waves become highly complex. So the organoid shows brain waves that are very similar to these premature human baby brains. And then, to ask if they are very similar to in age, we decided to create a machine learning where we use EEG data from several premature babies from 25 weeks to 38 weeks. We trained the machine to determine the age of the subject based on EEG features. And once the machine was really good, we start feeding the machine with the data coming from the organoid and ask if the machine was able to interpret or to predict the age of the organoid. And the data is just amazing. I'll show you. So the perfect data is going to be on the dot bar here. You have age in weeks, and you have the prediction age by the machine learning. So I hope you can see that the black dots are the real data. These are the data from the human brain. And the blue dots are the ones from the organoids. So they overlap. The machine gets confused. It cannot distinguish between the organoid and the real human brain. So that prediction happens after 25 weeks. Before 25 weeks, there is poor correlation. And there is one reason for that. We don't have any human data before 25 weeks. The human brain will just not survive. Right now, there are more and more neonatologists that are able to keep the human brain alive even at 23, 24 weeks. But this is really pushing the limits. Most of the premature babies before 25 weeks will just die. So we don't have any data to compare. So we cannot train the machine. So we still don't know what happens in the first 25 weeks of human gestation in the brain. So that data suggests that the organoids that we create in the lab follow the same trajectory in neurooscillation as the human brain. So you know what happens if you create an artificial network that mimics the human brain like that? If you're a scientist and you have a lab, you end up in the New York Times. So that was last year. We just finished that. We just complete that. So now we can recreate a brain organoid from any of you and understand how your brain oscillations actually happens in these very early stages of life. So one thing that I didn't mention is that after 9 months, the level of these networks stay kind of constant. It plateaus. And we were expecting them to continue to evolve. So we thought that the reason why these networks plateaus, it's because they don't have any stimulus. Right now, we grow these brain organoids inside an incubator, inside the lab, and they just stay there. Nobody is stimulating them. They don't see anything. They are not receiving any input. So That's very different when a baby is born. By 9 months, when you deliver it, the first thing that happens is the baby experiences the world, it cries, and then opens the eye and sees mom. And then mom sees the baby, and a strong connection happens. And the networks at that stage are just crazy for stimulus. So we are trying to mimic that, and one way to mimic that is to create a visual system. So what if we add the eye to the brain? So what you have here is 3 types of organoids. The first organoid is a retina organoid, where the photoreceptors are in green. In the middle, we have a thalamic organoid that receives the ganglion neurons from the retina and project to the cortex. And you can follow the trace of those neurons in red. So we have markers, so we know that we have the right structures. So these structures are now just maturing in the lab as we speak. And what we hope for is that they would mature to a level where the photoreceptors here would be stimulated by light, so we can pulse images, we can pulse light, and we can record from the cortex to see if the cortex now acquire a more visual identity. Then, if that's the case, we can ask questions like, can these brain organoids store a memory? Can we give them any kind of experience? So hopefully, we can cross that plateau activity and have the brain organoids to continue to evolve, so we can see different types of oscillatory waves appearing. So that's mimicking biology. But I was very anxious on stimulating these organoids, and I thought, what if we give them not only an eye, but an entire body, right? Why not, right? So we decided to create an interface with a robotic machine. So what you are seeing here is a robot that is learning how to walk by using the electrical information coming from one of these organoids. So the way this works is the following. The organoids are in the multi-electrode. We capture the electrical activity. We send the electrical activity to a computer who decodes that and tells to the robot how to coordinate the 4 legs. So that's what you are having here. Now we add those 2 eyes. These are not real eyes. These are infrareds. The infrared helps the robot to detect when it's approaching an obstacle, such as a wall. And as it approaches the wall, when it gets really close to the wall, it stimulates back the organoid. The organoid receives a new stimulus. It changes the network. The network will become something else. The computer will now realize that, well, the network changes. So now, instead of moving forward, we give a second command to the robot, such as stop and walk back. So the idea is to have these robots exploring the environment while they stimulate the organoid. And they are doing this all the time. So again, it's an artificial system that we are creating to stimulate these organoids and helping them to mature even further. We are also using this machine learning to create ways to understand how the neurons in these organoids self-arrange and understand how the human brain learns, so you can create better algorithms for artificial intelligence, for example. So this is a new partnership that we have with Microsoft. So that's one thing that I found it amazing, but there is another thing that I think these organoids are very useful, which is to understand our own oranges. So when I started my lab, the biggest question that I have was to understand what makes the human brain so unique. We are an outlier over all other species. So we are highly social, and we can create arts and technology, and no other species have done that. So why is that? Most of evolutionary neuroscientists, we are used to compare our brain with our closest primates, relatives, the chimpanzees. But again, I mean, when I look to a chimpanzee brain, I look to the human brain, they are so different that I cannot even compare. So instead, I was thinking about why not comparing the brain of our ancestral For example, the Neanderthals. And I don't know how much you guys know about human evolution, but there was lots of human types living on Earth at a certain time. The ones that we know the most are really the Neanderthals, because then we have lots of fossil records. So we learn a little bit about their lifestyle, how they live in community. But one thing that is, to me, most appealing is that even though they have lived on Earth for a period that is longer than what we have lived, their tools and technologies are limited. So someone might even argue that, whoa, there is a huge advancement here over millions of years, over several hundreds of years, but it's still a stone tool. We, modern humans, we changed the world, right? I mean, we start with stone tools, and now we have all these arts and technologies. We are here having this conversation because we learn how to live in highly complex society. We learn how to take advantage of human imagination. We learn how to communicate with each other in a very efficient way so we can advance all those things. And again, as I mentioned, no other species have done that. So, all right, so Neanderthals are like us, humans, so it would be better to compare their brains with the human brain. But there is a problem here. Although we can have bones from the Neanderthals, we can even extract their genome and sequence their DNA, we don't have live cells. And I need live cells to reprogram so I can make these brain organoids. So since we don't have live cells, we decided to take a genomic perspective. So we align the genome of Neanderthals and modern humans, and we ask, what is so unique about us? So if you look for the protein coding genes, which is about 20,000 genes, and you ask how many of those are different between us and the Neanderthals, we end up with 61 genes. And then if you ask, what are these 61 genes doing? You realize that there's a couple of them that are involved in the immune system. Some of them are involved in bones. They have like strong bones, muscles. But what about the brain? How many of those are involved in brain development? And that's fascinating. It's only 4 genes. So if it's only 4 genes that have point mutations that make us different from the Neanderthals, I don't need live cells from the Neanderthals. I can just edit a human genome to carry the ancestral genes of the Neanderthals. And then I can create Neanderthalized cells and create Neanderthalized organoids or Neandertoids, how we call them in the lab. So nobody in the lab wanted to do that because they thought, it's such a high-risk project. I mean, what if I do all this work and at the end there is nothing? So this project, I actually started myself. But finally, I convinced someone to help me, and the data pays off. So these are the organoids carrying the ancestral variants compared to the modern humans that I just showed you. So I hope you can appreciate how the morphology changes a lot. So this is all about how the progenitor cells migrate to form the microcircuitries of the cortex. And these microcircuitries are really important because when we measure their activity, they are very different from modern humans. So what does it mean? When we compare those networks, they are very similar with the networks coming from brain organoids from autistic individuals that we have in the lab. So, wait a minute, can you say that the Neanderthals are autistic? No, we cannot say that. That would be like a huge extrapolation. But we can say that there is something about autistic neurodevelopment that might be part of human evolution. It could be hyperfocus. It could be lack of socialization. It could be difficulty with language. So probably those 4 genes have helped us on our evolutionary path. So now think about it. If I using these organoids and genome editing to look back into our past, why not looking to our future? Why not manipulating the human genome to create other kind of brains that never exist, that evolution has never had the time to do it, so we can accelerate human evolution in a dish? So that's what we're doing. we can speed up human evolution. We are looking for regions in the genome that are highly polymorphic among us and create very different brain organoids. So what kind of brain organoids are you creating? We are creating brain organoids that are probably, that's going to be probably resistant to Alzheimer's. We are creating brain organoids that will have more synapses. We are creating brain organoids that are more plastic. So, I mean, these are all useful things. And as we reach to that level, we as a society, we have to decide if those genetic variants should be passed on to everybody. So that is starting to create some ethical dilemmas, and we can talk about that later. But this is the kind of project that makes me think about other futuristic projects. For example, last summer, we sent these brain organoids to the space station. So they were growing and developing inside the space station. And we were inspired by this program from NASA, who compare genetically identical twins, that one stay on the space station for about a year, the other one stay here on Earth. When he came back, they did the comparison and they realized that there are several circuitries or systems in the body that was affected. Some of those changes are reversible, others are permanent. Things like cognitive decline are very important for NASA because, I mean, if you are planning of sending someone to Mars, you need the astronaut to have a really good mental health. Problems with the retina, so the astronaut must see what's going on. So we thought that perhaps we have the right system to help on space exploration. So we had to build a machine that creates these organoids. So this is the kind of machine we have. It's a shoebox. And the organoids grows inside those tubes here. And we keep all the fluid that's coming from these bags and keep feeding the organoids as long as we wish. So we went to Cape Canaveral. We put everything in a box. You get the seal of approval from NASA. We put in a SpaceX. We hope it launches, and if it launches, you celebrate with your friends. About 32 hours later, I mean, the mechanic Canadian arm from the space station will get the capsule, the Dragon capsule, and we'll put in the space station. And the only thing that the astronaut has to do is to take that shoebox, put in a platform like that, and plug in one of those compartments. As soon as the astronaut plug, I receive a ping on my computer saying that it was plugged, so I have immediately access to everything inside, so I could monitor how the organoids were growing, how was the temperature, things like that. I could even take a picture. So this is one of the pictures. We need to improve the camera, by the way. But nonetheless, I mean, I hope you can see some spheroids in there. So these are the brain organoids. And there's a couple of things that we are learning. For example, look how perfect spheres they are. So when we grow organoids here on Earth, they are always slightly different. They're not perfect. But microgravity makes them really perfect. They are perfect spheres. Also, it seems like they grow a little bit bigger. So what is the consequence for space colonization? So if you have a baby in space, you probably need a C-section because your baby is going to be like a big brain. That is true. It's so funny because I never seen a science fiction book that talks about delivering a baby in another planet. But that's a real problem. I mean, I should talk to Elon Musk about that. But anyways, so yeah, so these are important considerations for astronauts and long-term space flights. But we also want to use this technology to learn something about human health here on Earth. And one thing that we and others have observed is when you bring those cells back, there is something really funny that happens, which is accelerated aging. Yeah, while in microgravity, the cells are beautiful, nice, but as soon as you bring them back to Earth, in about 2 weeks, we have this speed up, accelerated aging, so the cells just age faster. Nobody fully understands the mechanism. By the way, this is an open field. But we know it happens. So perhaps now, can I use these brain organoids to model diseases of late onset so I don't have to grow these organoids for several years to see, for example, plaques in an Alzheimer's brain organoid? Maybe I can just keep them over there for a while, then bring them back so I can study dementia, Alzheimer's, Parkinson, things that requires a long time to develop. Another consequence of this project was that as soon as we have those shoebox and growing the organoids in a fully autonomous ways, I mean, people in my lab start saying, why do you need me to change the medium? Just buy one of those boxes for me. And they are right. Because we keep doing these tissue cultures. It's like a 100-year-old technology. But what if we have all these boxes growing all these organoids for all the different projects? So the landscape of my lab, of how we do science, is changing. So we think that in the future we're going to have these brain farms. Lots of those boxes growing different brain organoids. And I tell you what, when I show you that experiment from cancer, so cancer centers start to pay attention on that, and they realize, maybe we also need our own brain farms so we can grow these organoids from our patients and test the different drugs before we test in the patients. So that might speed up all the clinical trials, so it might save lives. So this technology might be very transformative in that sense. All right, so I think I talked too much already, so I'll stop here. Just to acknowledge people in my lab, so these are folks who actually do the work, except for the Neanderthal thing that I started. But most of the time, I mean, they are the ones that do the work. The families that contribute to my research, and funding agencies. Thank you very much.
Yeah, so actually what I think we should do, this is the exploratorium after all, so why don't we show an organoid?
Yep.
So let's go to the microscope, as they say. So Dr. Muotri brought these up with him on the plane in his pocket, right through TSA, right? And what we're going to do is get them focused and start to look at a real organoid.
So I have a couple of them here. This is a small one. So these are about 6 months of age. So they grew for 6 months. It's a little bit dark inside because 6 months is already like kind of old. I hope what you can see is there are some structures inside, and these are the ventricular zone that I show you, in the neurons that migrate out and form the cortical plate here. So there are some of them that are better to see than others. Eventually, and I remember seeing one of them in here, we end up with structures that are, or cells that are very dark inside these organoids. These are unintentional retina cells. So sometimes the brain and the eye, they develop more or less the same time, and we end up with some photoreceptors in there, but unintentional. Most of the time we can grow them in a very simplistic cortical fashion. So yeah, progenitor cells will be inside. So as we see that it becomes darker in the center, it means that in the center we are not receiving the nutrients anymore from this media. So this is a media that contains all the nutrients and factors that these cells need to be alive. And that's why they stop growing. So they reach about 0.5 centimeter and then they will stop grow. There are some people in the lab that are creating a vascular system, so hoping they can at least double in size. Here you are seeing about 2.5 million neurons in here, plus all the glia cells and progenitor cells. And,
you were saying that's like a bee.
Yeah, so 2 million neurons is about like the brain of a bee. Yeah.
And how many neurons in a human brain?
The human brain has 86 billion neurons. Yeah. So what's fascinating, you might ask, come on, I mean, you just showed that those million neurons are similar to billion neurons. And that's one of the fascinating things that we are learning, that the brains, to form these neural oscillations, we don't actually need that many neurons. So you can do it with 2 million neurons. And I think when we think about consequences for example, neural networks for artificial intelligence, that's good news. So we don't need that many inputs or nodes to create these kind of networks. Let's see if we can find something else.
So one of the things that's fascinating to me is, what really are these things? I mean, they're not brains. I mean, it's kind of an interesting question, particularly when you get into ethics. What is this and what isn't it in terms of an object, a biological object?
Yeah. So we got, was so funny because we were having this discussion with different philosophers, and that question appeared. What are those things? And those philosophers, they said, they are not in the philosophy books. So it's something else. We have to create a new way to understand them, or to give them some kind of status. Are they humans? Are they cells in a dish? What are those? So that's open question. And I think I was joking with them that I carried those organoids in my pocket in a tube like that. We are having like lots of ethical and philosophical discussions related to organoids becoming conscious or self-aware. And really, there is no way for me to know. But let's suppose that in the future, we will figure out that they are. So it might be that in the future, I might not be able to do that. That might be like a crime to keep them here without the proper conditions. Because they will receive a moral status similar as an animal or a mouse in the lab. So that's, that might be the future. Yeah.
So you're using this as a model really to learn about the brain as well as to do some biological work. How do you think about the fidelity question? I mean, how do you deal with the fact that this is, I mean, you saw those the electrical signals coming together, which must have been very exciting, by the way. I mean,
yeah.
That must have been an extraordinary discovery. How do you tease and figure out, well, how much of this is really something unique to this kind of object and something as opposed to something about the brain itself? We're on a trajectory of development. I mean, are there some artificial things that you're seeing that you have to worry about that may not be true in terms of the real brain development?
Yeah. So I think there are two possibilities, right? One is we are mimicking human biology in a very precise way here. And we have evidence that that might be the case, right? The trajectory is the same, the cell types, the identity, so they're doing what they're supposed to do. It seems all genetically programmed to do that. But on the other hand, we know that these are growing outside the body. They are not connected with other brain regions. They are not in the uterus. So they probably are not doing what they are supposed to do because they don't have all the right conditions. So I think scientists now, like me, I mean, we see things that are similar and we see things that are very different from the human brain. It might change in the following years as we improve the technology. Those things will become more and more close to the reality, which is the human brain. But I think the important take-home message here is that even though if they're not similar, even if we already have, we can learn so much about the human brain, how it forms in that very early stages, how the first neuron polarize, how the first networks start to get together. That I think it's a tool that in the future we're going to be looking back and said, oh my gosh, how did we survive in neurosciences without this tool?
So you have such passion about this work, and it's interesting to think about what's really driving you to do this research. I mean, what's really, you're doing so many different things here. How would you describe what's the inner drive for this? What's making you want to do this?
Speaking as a scientist, I think it's really the possibility of discover something that nobody has seen before. I mean, we all have this ego of being the first, of doing something. This is part of the science, part of the motivation. I had that at one point. Now, I think in a different way. I have a kid with autism, very severe autism and epilepsy and seizures, and there is no medication, there is nothing that can help him. So I think this is a tool that we need to attack or to improve the life of people with neurological disorders. And when we think broadly about neurological disorders, we are talking about millions of people. So I think that's my main motivation, is the translational aspect of that. How can I move from this very simplistic model to helping people with neurological conditions?
So I have one more question, but before I do, I want to put an ad in for people to go and look at the exhibit that's in the back part of the museum, Cells to Self. This lecture series is really to talk about some of the current breaking edge science that's happening about that same topic that you can't really show easily in an exhibit, although pretty soon I think we can have an organoid in our exhibit. So go and see that exhibit and then come to these other shows. I think my final question is a little bit something we talked about earlier, which is, why is it so critical for people to learn about this? I mean, this is kind of a pretty advanced scientific topic. But you said this is really something that one needs to know about deeply. So what are your comments on that?
So, and that's why I applaud your initiative to have these conversations. I've been in the stem cell field for a while, and I remember when, I mean, scientists cloned Dolly, right? I mean, we cloned an animal. Large animal, and everybody was taken by surprise. I mean, you could see people freaking out. So now it's Dolly, tomorrow it's going to be a human. So how come? I mean, we are cloning people already, and but nobody was informing the community or the society that of the scientific advancements that was happening along the way to get to that stage. And I think if we know everything that happens along the way, we, as a society, we are much better informed of the uses and consequences of that. So we can support this kind of science, or we can control it better. So I don't want to have this technology to become the next cloning. So even the name is wrong. You see, I mean, we use clone, and it's not the proper scientific name. Here, people start calling them mini brains. It's a wrong name. Again, we are making the same mistakes again. So having this opportunity to really talk, and I think it's important for people to realize what is the stages, what is the possibility, what this technology can bring to the table that we never had before.
Well, with that, join me in thanking Dr. Muotri for a wonderful talk. And we encourage you to go out and have fun. We're still open till 10 tonight. Thank you for coming.